Okay, let's talk about some of the different fates of pyruvate, and in particular we're going to talk about the process of fermentation in yeast and in humans. So if we break down glycolysis, right, we start with a molecule of glucose, and through the first 5 reactions we convert it to 2 molecules of GAP. During that process we use 2 ATPs, and that's the energy investment phase. In the second half of the reaction, our 2 GAPs are converted and oxidized to 2 molecules of pyruvate. We use 2 molecules of NAD+, generate 2 molecules of the reduced cofactor NADH, and we form 4 molecules of ATP. Now, when we think of glycolysis, we said that the rate limiting step is up here at phosphofructokinase. So that's the slowest step, and that's highly regulated step because it can determine the speed of glycolysis. But what we haven't mentioned is what the limiting reagent is. Now the limiting reagent obviously depends on the situation. Sometimes you don't have enough glucose and you have to get glucose from other places like glycogen or from breakdown of proteins. So glucose can be a limiting reagent, but in the absence of oxygen, it turns out that NAD+ is the limiting reagent. So in anaerobic conditions, NAD+ is the limiting reagent. So if I held my breath and I went sprinting down the hallway, the first thing that would run out is my supplies of NAD+, right? Now, in the absence of oxygen, yeast and you have come up with sort of a side fate for pyruvate that can help replenish NAD+ to a certain degree, right, for a limited time. And the process in yeast is called fermentation. And so let's look and see how fermentation works. So let's draw out the structure of pyruvate, and we'll see what happens as we go through fermentation, which will be in blue here. Okay, it turns out fermentation has 2 steps. The first step, which is categorized or catalyzed by an enzyme called pyruvate decarboxylase. Again, these enzymes give us lots of clues as to what they're doing. This is a decarboxylase, and so it removes carbon dioxide. It converts pyruvate to a molecule of acetaldehyde. Okay. This acetaldehyde is converted by the action of alcohol dehydrogenase. Remember I said I was going to call these dehydrogenases DH, into this molecule here, ethanol. Okay. Now if you look at acetaldehyde versus ethanol, we've got an aldehyde with its carbonyl group versus an alcohol. This is a reduction reaction, right? The acetaldehyde, an aldehyde, is more oxidized than an alcohol. So we have reduced this carbon chain. And in the process of this reduction reaction, we're able to oxidize NADH back, whoop, let me erase that. That's not a great eraser. To NAD+ plus. And this is really the whole purpose of this fermentation reaction, is to generate NAD plus in order to feed it back into glycolysis so that glycolysis can continue for a little while longer. Since we run out of NAD plus, one thing we can do, or yeast can do, is to ferment its pyruvate to regenerate NAD plus, to pull it back in and replenish that limiting reagent. Okay. Now there's consequences to fermentation, consequences that we've exploited, right? This is how you make ethanol, right? If you want to brew beer or make wine, you take a sugar source, grapes, grain, whatever, you put it into a fermentation vat, which is sealed off from oxygen so that you have anaerobic conditions, and you contaminate it with some yeast. That yeast will metabolize that glucose in order to make more yeast cells, in order to do its metabolism, in order to generate those ATPs, and it will break that glucose into pieces through glycolysis, eventually getting running out of NAD plus, the limiting reagent. So in the absence of oxygen, it will start fermenting its pyruvate to generate the bubbles that you see in beer and alcohol. Now, the problem with this for yeast is it can't go on forever. Eventually, ethanol is toxic to yeast or bacteria or to us, right? And yeast can only produce around 5% ethanol before they stop dividing and stop metabolizing. Now, in order to make more higher concentrations of ethanol, like in liquor, you have to do a distillation on that low percentage of ethanol that was formed in that first fermentation from the yeast. So this is how we make booze, right? We seal up some sugar source that has yeast in it and ask that yeast to generate ATPs in the absence of oxygen. Okay. Now, what do? Let me get a different marker here. We have the same problem, right? If we want to continue burning glucose to generate ATPs in the absence of oxygen, we're going to run out of NAD+. And we don't make ethanol. We make this molecule here, which you probably have heard of before, right? This is our lactate from lactic acid. And again, what we're doing is we're taking an oxidized carbonyl, reducing it to an alcohol, and so this reduction can be coupled to an oxidation of NAD+, or of NADH, NAD+. This NAD+ can go back and replenish this limiting reagent from glycolysis. Okay. Just like in yeast, there's consequences to lactate, right? This is acidic. We can only tolerate certain concentrations of lactic acid before we become acidotic. Our pH decreases and we start shutting down, just like the presence of ethanol starts shutting down yeast. Okay? But in the absence of oxygen, fermentation or lactic acid production are invoked as fates of pyruvate in order to replenish our limiting reagent NAD+, so glycolysis can continue. Okay, in the presence of oxygen, there's a much better fate for pyruvate, which is the Krebs cycle, which we'll be talking about very soon. The Krebs cycle is able to convert NADH into NAD+ and couple that electronic potential energy to ATP production and regenerate NAD+ so that we can continue to burn glucose. Okay, but we need oxygen in order to be the final electron acceptor of these electrons. Without it, we have these sort of rigged systems that only work for so long.